Hello and welcome to this SKC training event on sampling for contaminants of biological origin. I am Debbie Dietrich, the Corporate Industrial Hygienist for SKC, and I am pleased to present this webinar. When I use the term biological contaminants, I'm sure what comes to mind for many of you is toxic mold, oh my. Several years ago in the U.S., toxic mold was featured almost daily in newspapers and television shows as a major public health concern in homes and commercial buildings. Biological contaminants, however, include much more than just mold. So let's define the issue. Biological contamination is defined by the American Conference of Governmental Industrial Hygienists as aerosols, gases, and vapors of biological origin of a type and concentration likely to cause disease or adverse health effects. Contamination includes inappropriate levels indoors of biologicals typically found outdoors, or actual biological growth indoors. Now, it is normal for biological particles found outdoors to be detected indoors at low levels, and then we drag them in on our shoes. But growth indoors is never acceptable. Most biological contaminants are aerosols. In other words, they are airborne particles, including bacteria, fungi, pollen, and viruses. Biological aerosols may also include chemical byproducts, including endotoxins and mycotoxins, as well as other fragments such as skin scales. There is a great TV commercial in the U.S. that says, in 8 years, the weight of your mattress doubles due to skin scales and dust mites. Now there is a good sales pitch. Biological contaminants also include vapors. We all know that musty smell of mold in a locker room or basement. This is technically a microbial VOC, or MVOC, produced as a byproduct of microbial metabolism. So let's look at what bacteria industrial hygienists may possibly encounter in the workplace. First, Legionella, a bacteria that can grow in water systems, including cooling towers. Pseudomonas, a bacteria associated with metalworking fluids. And finally, Staph aureus, including the methicillin-resistant superbug MRSA. Workers can get the infection from another source, like a hospital procedure, and then spread it through skin-to-skin contact in the workplace. Some so-called gram-negative bacteria may also contain harmful chemical substances called endotoxins. Note that gram-negative bacteria are those that do not take up the standard microbiological gram stain, or dye, if you will, used in microscopic analysis of the organisms. Note also that endotoxins are sometimes called pyrogens, as they induce fever. SKC does not sell so-called certified endotoxin-free filters. Instead, professionals can run blanks of standard filters for different analytical procedures. Polycarbonate filters are typically used for endotoxin analysis using a procedure called the LAL assay. If you read about endotoxins in industrial hygiene reference books, exposures are typically found in industries shown on this slide: cotton textile mills, poultry and swine operations, and in other operations with metalworking fluids. Exposures can be found in the general public anywhere where there has been a water intrusion event. The primary biological contaminant implicated in indoor air complaints is mold, not such a fun guy after all. Shown on the slide are some species of mold commonly found in indoor environments. Now I would say mold are like people. They need food and water. And they can eat any non-living organic material like wallpaper or sheetrock. Give them a little water and they have a mold champagne brunch. The term toxic mold is used to describe not these common everyday molds, but those molds that produce a chemical substance called a mycotoxin. So to clarify, bacteria produce endotoxins. Molds produce mycotoxins. Mycotoxins are only produced by some species, and their structure and function are quite diverse. In the workplace, mycotoxin exposures typically result from inhalation of fungal materials from water-damaged materials. In the general community, mycotoxin exposures typically occur from ingestion of moldy foods. Some mushrooms, for example, may contain mycotoxins, which is why you need an experienced picker if you're getting them from the woods. So let's now look at who are the workers who could be exposed to biological contaminants. In general, workers potentially exposed to bioaerosols are those who come in contact with plants or animals in their job, including agricultural workers and food handlers and processors. Other workers potentially exposed include industrial workers in industries like pulp and paper mills, textile mills, wastewater and sewage treatment, and again, any industry with a cooling tower. Healthcare workers, remediation workers, and office workers with humidified indoor air are also potentially exposed to biological contaminants. Now this is a presentation on sampling, but I wanted to give you some idea of the health effects of these contaminants. Now some professionals may question the need to sample for biological contaminants, since there are no regulatory exposure limits. Well, typically, professionals do air sampling because people are sick now. Sampling is done to identify the root cause and to develop corrective actions, not for compliance with a limit. In the workplace, you may see skin infections caused by biological contaminants, things like MRSA, ringworm, and rashes. Of course, the most common health effect is eye, nose, and throat irritation, the so-called sick building syndrome. Biological contaminants may also induce fevers, like humidifier fever from endotoxins or Pontiac fever from Legionella bacteria. Biological contaminants may induce allergic reactions or so-called hypersensitivity diseases. Many people exhibit hypersensitivity symptoms to dust mites. In the workplace, hypersensitivity diseases include a pneumonia-type illness and building-related asthma. It's worse on the weekdays and improves on the weekends. We've all gotten sick from somebody else's biological contaminants when they sneezed on us or gave us a contaminated handshake. But some really serious infectious diseases can be found in the workplace, including Legionnaire's disease and TB. Note that oftentimes these health effects occur quickly, not like asbestos that causes health effects 40 years after exposure. So again, professionals do air testing for biological contaminants, not for compliance, to exposure limits, but to get their people healthy and back to work as soon as possible. I once read that mycotoxins are among the most toxic chemicals on the planet. In short, mycotoxins will zap your immune system, which will increase your susceptibility to disease and will reduce your ability to fight these infections. You can see on this slide the wide range of health effects from mycotoxins. A very notable case occurred in Ohio when children in a daycare center experienced pulmonary hemorrhage, and mycotoxins were determined to be the probable cause. Another case on the TV show 60 Minutes featured a home that sustained significant flood damage. The resultant mycotoxins led to various health effects on the brain and nervous system of the occupants. So that's why professionals do their testing for biologicals. Let's now look at how to conduct the investigation. ACGIH recommends a stepwise approach with these 4 critical steps. Step 1, gather information, do interviews, surveys, inspections. Then formulate a hypothesis. What do you think is causing the problem? Then collect air samples for the express purpose of testing your hypothesis. And finally, make recommendations for controls. Let's now look at the information gathering step. In summary, if you get a complaint about indoor air quality, do not rush to collect samples willy-nilly. Instead of reaching for sampling equipment right away, first reach for a clipboard and note things like complaint versus non-complaint area, date when problem first noted, days or times when problem is more or less, and finally seek input from the building occupants so you can formulate a hypothesis on the root cause. In the information gathering step, do a building inspection, examine the physical structure, maintenance, and occupancy patterns. Look for potential sources of biological contaminants. In particular, look for moisture indicators such as watermarks, visual presence of mold, that musty smell. And this last one was new to me, and I may be new to you as well. White powdery or crystalline substances on the surface of concrete, plaster, and masonry. These are soluble salts dissolving out from the building materials. A moisture meter is a critical tool in mold inspections. A moisture meter can be used to survey moisture in a variety of materials like ceiling tiles, gypsum board, wood, and plaster. Shown here is a moisture meter from SKC. It operates by measuring the conductance between those 2 probes. This style of moisture meter is best used in construction or renovation projects when the test surface can be punctured. In regards to data interpretation, moisture levels can be compared from wall to wall to figure out where moisture intrusion is occurring. Once you find the source of the moisture, an investigation can be made as to the cause and a control strategy can be developed. The Western Wood Products Association also gives some guidance to aid in data interpretation. They recommend that the moisture content of lumber be kept below 20% to prevent the growth of mold. GreenGuard Environmental Institute has received ANSI approval for a standard covering the management of moisture and mold growth during construction. See greenguard.org for complete details. Now, during the building inspection, you want to do an HVAC system check. That is to ensure the outdoor air supply meets ASHRAE or other appropriate standards. Also check the location of the air intakes. Air intakes on rooftops can draw in biologicals from cooling towers, sanitary vents, building exhaust, and animal waste like pigeon droppings. Air intakes at street level can draw in moisture, vehicle emissions, and odors. The condition of the air filters is also critical. Filters may themselves promote the growth of microorganisms if they become wet. Finally, check the supply air. Ductwork should not be coated with excessive debris. Also ensure cold air leaving a diffuser does not produce condensation and the potential for microbial growth. Next, we will discuss how to choose a sampling method to test the hypothesis, along with some data interpretation tips. Sampling for biological contaminants is done to test your hypothesis, to positively confirm the absence or presence of biologicals, to identify the type of microbe at the genus as well as the species level, and most importantly, to confirm the effectiveness of decontamination. The mold spore count, for example, before and after remediation can be quite compelling and convincing data to show to affected individuals. Sampling is done using either bulk, surface, or air samples, followed by analysis at a qualified environmental lab. Now, with bulk sampling, portions of materials in the building are tested for mold or other contaminants. Things like wallpaper, a piece of carpet, the return air filter, duct line, even settled dust. Portions of the test material, like carpeting, are placed in a Ziploc-type bag for transport to the lab. In some cases, you may instead use a sterile jar for something dry or a sterile bottle for water, if you're like looking for Legionella, for example, or for metalworking fluids. Settled dust in a room can be collected with a standard vacuum cleaner, just using a new vacuum cleaner bag for each sample. Next, let's discuss surface sampling. Carpeting is an ideal test surface. It's just sitting there. It's an effective reservoir for fungal spores and can reveal the history of mold in the building. To sample fungal spores in carpeting, we typically use a vacuum-style cassette. What I'm talking about here is a standard 3-piece closed-face cassette, but on the inlet, you insert a little piece of tubing to serve as a nozzle. And with mold, we typically use a 0.45 micron polycarbonate filter. We sample at high flow rates, really digging that piece of tubing, that inlet, into the pile of the carpet as deep as possible to get a good air sample. On this slide, you can see the micro vacuum cassette in the SKC carpet sampling kit. We also supply the little template so that you are vacuuming up a consistent test area that allows you to better compare the results. This slide offers some guidance on data interpretation. Normally, you should see less than 5,000 spores per 1,000 square centimeters. If you're at 75,000 per 1,000 square centimeters, you have elevated levels. Sterile wipes are also used for surface sampling. In this case, a swab or filter wetted with sterile water or other solution is used to wipe the specified area. Then the sampler is used to inoculate an agar plate for growth culture. This technique is used for MRSA testing. This slide offers some guidance for data interpretation. Normally, you should see less than 1,500 CFUs, colony-forming units, per square centimeter. Greater than this, probable contamination is indicated. Now, it may seem very low tech, but lift tape is a common method for surface sampling. And what we're talking about here is samples are collected by placing clear adhesive or regular old packing tape onto a surface and removing it with slow, steady force. Then you put the piece of tape onto a glass slide and it is examined microscopically. SKC offers flexible plastic microscopic slides with a sticky adhesive sample area, and we call these slides stick-to-its. You simply press onto the test surface, then put the slide into the plastic mailer provided, and send it to a qualified lab for microscopic analysis. Here you can see some data interpretation guidelines for lift tape samples. Normally, there should be no significant fungal material. Contamination is indicated when the sample contains 25 to 100% spores. Like with chemical sampling, air sampling for biologicals is done for the purposes of evaluating actual human exposures. And air sampling for bioaerosols may involve the use of impactors, filters, or liquid-based impinger-type devices. Air sampling for mold is often done with spore trap cassettes. Spore trap cassettes are easy, inexpensive screening devices used with pumps that flows at 15 to 30 liters a minute for very short sample times up to 10 minutes. They resemble a standard filter cassette, but there is no filter inside. Instead, the collected spores impact onto an internal microscopic slide with a sticky surface. Then the slide is stained and analyzed microscopically by the lab. Here you can see the SKC spore trap cassettes called VersaTrap and a diagram of the airflow. SKC VersaTrap cassettes are versatile samplers that allow for flow rates up to 30 liters per minute. This enhances the collection of smaller mold spores compared to other brands of spore traps. The SKC QuickTake 30 is the perfect partner for spore trap cassettes. The QuickTake 30 features constant flows up to 30 liters per minute, user-selectable programmable sample times, lithium-ion battery, and indefinite runtime from the AC adapter. Spore traps provide a lot of good information for a relatively low cost. For example, they will tell you the total number of spores and the type of the spores at the genus level. This information can be used to compare the complaint area to the non-complaint as well as to the outdoors. The genus of the spores should be similar inside and out, but the numbers should be lower inside. Shown on this slide is data interpretation guidance using spore traps. In a residential structure, normally you should see less than 5,000 spores per cubic meter. In a commercial building, it should be less than 2,500. And that's because a higher standard of care and cleanliness is expected in commercial buildings. Globally, the gold standard for sampling and analysis of biological contaminants is a viable cascade impactor. This sampler is specified in NIOSH methods for bioaerosols. Shown here is the SKC viable cascade impactor called the biostage. As per method requirements, it is used at 28.3 liters per minute for sample times up to 5 minutes. Bioaerosols impact onto growth medium or agar supplied by the microbiology laboratory based on the target contaminants. Here you can see SKC biostage sampler preparation with the agar plate. The QuickTake 30 sampler pump has a mounting bracket to attach the biostage sampler for easy transport. Also note the valuable sample assurance tip shown on the slide. Be sure to clean the impactor with isopropyl alcohol before each use and evaluate blank samples of your agar plates. Standard filters, including polycarbonate, MCE, and PVC, are also used to collect bioaerosols. Sterile gelatin filters sold by SKC, however, are often a better choice. These filters help to maintain viability by minimizing dehydration of the microbes. Gelatin filters can be used with the SKC button sampler or other samplers. An AIHA journal article reported that 25mm filters used with the button sampler provided collection efficiencies close to 100% for airborne spores. If you want to collect viable samples for growth culture, filters, support pads, and cassette or holders should be sterile. Samples are collected with a portable pump at 1 to 4 liters per minute for, again, short sample times, up to 30 minutes, to ensure viability. Another advantage of the gelatin filters for growth culture is they will dissolve when they're placed onto the agar. Bioaerosols are often collected into a liquid medium using an impinger-type device. Liquid collection allows for a number of different analysis techniques. Shown here is the SKC biosampler that is normally used with a microbiology-grade mineral oil called SKC Biotrap. The advantage of the SKC biosampler with Biotrap is that sample times up to 8 hours can be used. The other methods we discussed so far in this presentation had much more limited sample times. With the SKC biosampler, particle collection is achieved by drawing the aerosol through 3 nozzles directed at an angle towards the inner wall. This creates kind of a swirling action, like a washing machine, and gently removes collected particles. Again, this gentle swirling action helps to ensure viability. Analysis options with the biosampler are shown here. As always, with any analysis, check with your lab before collecting the sample to ensure they have the capability of providing the analysis you need. I'll show you here are 2 common questions we get all the time at SKC regarding the biosampler. Why is the sonic flow pump required? Can't we just use the QuickTake 30? And secondly, can the biosampler be used for PCR DNA-based analysis? Now, regarding question 1, the biosampler was researched and validated using a sonic flow pump. Sonic flow pumps provide a half an atmosphere or 15 inches of mercury pressure drop. This pressure drop causes the air to flow at the maximum speed, the speed of sound, which enhances collection. If you use a pump that does not provide this amount of pressure drop, like the Quick Take 30, you just won't be as efficient in collecting small particles. But many customers do use the Quick Take 30 as a more convenient option. Regarding question 2, polymerase chain reaction PCR analysis requires the use of water, not the SKC Biotrap, which is mineral oil. You can indeed use water in the biosampler if you wish, but water may evaporate over the sample period, reducing sample times to less than 8 hours. Shown here are data interpretation guidelines for viable air samples. Residential buildings should have less than 500 colony-forming units per cubic meter, and again, commercial buildings, less than 250. Air sampling for microbial VOCs is not normally done due to technical difficulties in analysis and data interpretation. If desired, specialty sorbent tubes or stainless steel canisters can be used. Some professionals actually may choose screening devices, including photoionization detectors or a mold dog. Finally, let's look at some general guidelines for data interpretation. So what about exposure limits for biologicals? There are few established standards except for things like wood dust, cotton dust. And ACGIH actually indicates that a general exposure limit for biological agents is not scientifically supportable. Why is that? Well, biological contamination is a complex mixture. Health effects vary. There is no single sampling method that is appropriate for all types of biological contaminants. And there is insufficient evidence to support a dose-response relationship. So, what do? ACGIH recommends gather the best data possible and use your own best judgment. In general, fungi found in indoor air should be the same species as that found in outdoor air, but the levels should be lower. If there is a dominant fungal species indoors that's not present outdoors, where did it come from? It's growing from some source within the building. Comparisons of indoor-outdoor, problem, non-problem areas should be at the species level, not at the genus level for the best information. Some species like E. coli may be considered indicator organisms. In order to compare the results from different zones, use an identical sampling protocol. And finally, how do you know when you've finished remediation? Primarily, fix the water problem. You then address your mold issues. And finally, no health problems on re-entry. Thank you very much for your attention. You are now an official SKC mold buster.